All right, we're back to wrap up the lecture for July 10th. Thank you. Great. We have been talking about proteins. All right. Protein structures. We went through primary, secondary, tertiary, quaternary structures. All right. Now, let's look at how you get a protein that functions differently. A change in sequence of amino acids, a change in the primary structure, leads to a different folding pattern. So the secondary and tertiary structure of the protein looks different. This can mean that the quaternary structure is then affected as this subunit or this single polypeptide bonds with other polypeptides to make a fully functional protein. All right? So let's look at the example of sickle cell anemia or sickle cell hemoglobin, which leads to the condition known as sickle cell anemia, right? In normal hemoglobin, you have this structure of amino acids, right? That folds up, makes a secondary tertiary structure of the beta subunit right here. If you remember from the previous slide, alpha, beta subunits in a normal hemoglobin. All right? The molecules function normally. They don't click together. They don't associate with each other, and they carry oxygen through the use of basically a cofactor of iron and heme molecules here. All right? And you get a red blood cell with this shape. A single change in the amino acid sequence can lead to an exposed hydrophobic region, all right, on the beta subunit. And that hydrophobic region wants to interact with other hydrophobic regions. So you get a sickle cell hemoglobin, you get a bunch of these, and now all the hydrophobic regions, they just pop together because that's what hydrophobic regions do. Like dissolves like, you know? They form together into these chains. They sort of crystallize into fibers, as put right there. They're not as good at carrying oxygen, and they give the red blood cell a specific shape that looks like a sickle. All right? So here's a sickle cell right there. And sickle cell anemia results from the reduced ability of these cells to carry oxygen. Additionally, these cells themselves can sort of hook together, and you can get more blood clots. But individuals with sickle cell blood cells, all right, they're more resistant to malaria, something that you're probably familiar with hearing about. So in areas that have high rates of malaria, the rate of sickle cell anemia is higher, okay? Or the rate of sickle cell genotypes is higher because those individuals are better at dealing with malaria. So it's very interesting. You have two types of selection going on, two competing selective pressures. Now let's dial it back to the column that isn't included here, and that's the sequence of the DNA. Because remember, the DNA encodes this sequence of amino acids. So a change in sequence of DNA leads to this change in the sequence of amino acids, leads to this change in blood cell shape, leads to a change in the whole organism's phenotype. All right? I now get anemic more often and I'm more prone to blood clots, but I am resistant to malaria. I have a new phenotype, all because of a very small change in DNA sequence. And then that can be passed on to offspring, right? Natural selection can operate, and that can become more or less prevalent, something that we'll talk about near the end of the course when we hit our section on natural selection and evolution. Okay, a protein's shape determines its function. A protein's shape can be lost. If a protein loses its shape, it is no longer going to have the same function. Proteins losing their shape, we call it denaturation. Denaturation is not a protein-specific word. It's used for all sorts of molecules. When you denature something, you remove its nature, all right? You get rid of its shape. So how can we do that? Well, let's use my arms again as a long polypeptide chain. I've got positive, negative, ionic bond. I've got hydrophobic, hydrophobic, hydrophobic interaction right here. That's giving me shape, right? And that's pretty good. But now you start to heat me up, all right? You put me in a frying pan, and now the molecule starts to move. And hydrophobic interactions and ionic bonds aren't as strong as covalent bonds, so they just go flop, and then the whole thing just keeps sort of wiggling. And now the protein's been denatured. Now, will it naturally go back to its original shape? Not always. Sometimes proteins can be renatured, but that's less common than proteins remaining denatured. Now, the main ways that proteins are denatured that we talk about are heat and acidity, all right? Or extreme pHs, because a basic solution does the same thing. Imagine that this interaction between my hand and my shoulder is an ionic bond, and I dump a bunch of hydrogen ions here. Well, I've already been giving you this information in previous lectures, but my negatively charged shoulder, well, now hydrogen gets in there and neutralizes that negative charge. So now there's nothing holding this together. The hydrophobic interaction here going on between my hand and my head, all right, that isn't maybe affected as much by the extreme pHs, but remember that this tertiary structure has a bunch of different side chain interactions. And the hydrogen bonds, those are certainly going to be denatured or broken down by an extreme pH, all right? Yeah, so you see how an extreme pH can interfere with those side chain interactions, causing the protein to lose its tertiary structure. It can also interfere with the sorts of interactions that give the protein quaternary structure when those two different subunits are joining together. It's usually through the same sort of, I have hydrophobic and hydrophobic domains, right? Or I have a positively and a negatively charged region. It's all the same sort of stuff. All right, cool? So extreme pHs interfere with some of those. You heat something up enough and its initial structure, it starts to jiggle. It moves around enough, the bonds break, and they can't always go back. Make sense? Cool. pH, temperature, ionic strength, which is, it's the same as the pH discussion of, hey, the charges get neutralized, right? Because ionic strength is just particles that have charge, ions, all right? It's the same principle as the pH. But you can use enough salt, you can denature a protein. All right, because you interfere with the bonds. Is this working for people? Now remember, folks, remember, this is tricky. This is tricky right here. So let's look at this real quick. Let's sit back and look right here. The bonds that hold the backbone together, okay, these are all covalent bonds. Covalent bonds are an intramolecular force. They are much stronger, much stronger than any of these sort of intermolecular forces that we've been talking about, such as a hydrogen bond, all right? Or a hydrophobic interaction is also much weaker. So when you heat up a protein or you expose it to extreme pHs, it's probably not going to affect the backbone. What it will affect is the tertiary structure pretty rapidly. And then if it's strong enough to break the hydrogen bonds, which are the strongest of the intermolecular forces we've been talking about, okay? If it's strong enough, bless you, if it's strong enough to break the hydrogen bonds, did I just ruin your sneeze? Sorry, that was an evil thing I just did to you. No, it's not, it was bad. If the extreme temperature or pH is strong enough to interfere with hydrogen bonds, then you lose the secondary structure, okay? So, as you expose a protein to extreme heat or extreme pH, it loses its quaternary structure first, then its tertiary structure, then its secondary structure, because the hydrogen bonds are consistent. They're the strongest of the interactions they're the strongest of these sorts of interactions forming these levels of structure. And then eventually, if you get something really hot, yeah, the covalent bonds will start to break. But that's extreme. Question?
Is that the order in which it goes for all protein denaturation, or just ones caused by pH?
I am positive that there are examples of proteins where the quaternary structure is more stabilized than the tertiary structure of each subunit. But the rule of thumb is definitely that you lose the quaternary structure first, which makes sense in like a metaphor or an analogy, right? Of you think about all the different subunits of, say, a car that make the car go, and those different independent pieces are usually pretty stable, but the things holding them together can fall apart and break. It's all held together by, like nuts and bolts and plastic and stuff like that. So it's kind of similar there. In general, think of the tertiary structure as more stable than the quaternary. The secondary is more stable than the tertiary, which this should be a very consistent rule because the hydrogen bonds are very stable. And the primary structure is 100% the most stable of any of these because it's all covalent bonds along the backbone. And those are tens to hundreds of times stronger than the other types of bonds that are occurring here. Cool? In fact, I think I removed this from my presentation. In the fall, I always use this, but let's go with relative strength of chemical bonds. I want to find us a nice image here. It's usually, this is the one I like. This is the one I use. I'm just going to project this real quick. All right. So if van der Waals forces are given a strength unit of 1, remember those van der Waals forces are just due to random patches of positive and negative charge. Dipole-dipole forces, okay, are 10 times stronger than that. That's when there are two polar molecules that sort of interact. Then we've got hydrogen bonds, okay, 50 times stronger than the van der Waals forces. But then we've got ionic and covalent bonds being 1,000 times stronger than van der Waals, or 20 times stronger than the hydrogen bonds. All right? So this is kind of, I think, this is kind of nice. And you can, of course, you can find additional diagrams that break this down more into like the single, double, and the triple covalent bonds and all that, because sometimes you need to know what's going to break first. All right? But, yeah. There you go. Cool? Exciting. So let's look at the egg. Here's another picture of an egg. I have this idea for this course, if I ever have a semester where I'm not teaching 4 courses at once, where I want to do an entire month of Bio 105 just on an egg, because there's so many principles you can talk about with them. But for us, we just talk about it during the biological macromolecules protein section. Here's an egg, all right? Look at the new albumin. It's coming out soon. And you crack the egg, and that's like a funny joke that you can do if you want to break up with your significant other, because it's not a good joke. Anyway, protein denaturation. It's all around you. Here's albumin. This is protein, the clear protein surrounding the yolk of the egg. You heat it up, it denatures. It was water soluble, it becomes insoluble. All right? You fry the egg, the protein gets denatured. Milk. All right? This milk is having like a citric juice, a lemony juice added to it, and then it's getting heated up because that protein's pretty stable. It's more stable than the albumin. You can heat up milk and the protein doesn't really denature. You've had a warm glass of milk maybe at some point in your life. But if you go warm milk plus acid, that thing denatures and it curdles, okay? I always tell this story of when I was in middle school or maybe high school. I lived in a very small town, and a family friend was like, we got tickets to go on a wine train. You want to come on the wine train? And you go on a train and you get a tea service, and then they go to a winery and the kids hang out. And we played hacky sack, and the parents went and drank. Anyway, I thought it was fancy to put all the stuff they offer you in the glass of tea. So I make my tea, and then I add the lemon, and then I add the milk, and then I add the sugar, and then I stir it, and it curdled and denatured the milk, and it was like I had cheese floating around in my cup of tea. But I was too embarrassed to say anything, so I just drank the tea. And that's an important lesson in denaturation. All right? So another example of denaturation that you may be aware of is the production of ceviche. All right? Where you use a very strong acid, like some lime juice, you soak meat in it, and that meat, the protein, gets denatured. Denaturation tends to make proteins easier to digest, and extreme pHs can also kill bacteria that would otherwise be there. So, that's win-win. It's chemical cooking. All right? Questions about that? Yes. It was horrible. No, I hated it. I didn't know what was going on. I was like, there's something wrong with my tea, but I just didn't say anything. Yeah. It's also, I had a friend whose brother was a bartender, and they had, if you had a troubled person at the bar, you would give them a special shot. It was like, it's on the house. And it was like a cream-based liqueur, and you had to bite a lemon. So you hold it in your mouth, you bite the lemon, and then you swish your head, and it makes the cottage cheese in your mouth. And that was how they would get rid of people in that bar. It's fun stuff, but it's all basic chemistry and biology. Isn't that great? All right, I love it. Nucleic acids, the last group. Ready? We're going to do the last group of biological macromolecules, the nucleic acids. This is DNA and RNA. We're going to talk more about DNA and RNA because we've got a whole week of just genetics talk. All right? So, DNA, RNA, what do? They serve as a template for directing protein synthesis. That's the level of understanding we need for now. I'm going to add more details as we get later in the semester. Okay? So the DNA is a blueprint your body can read to make some RNA, which is another blueprint your body can read to make a protein. All right? Why is there the intermediate step? Because there's a bunch of other types of RNA. Don't worry about it. I just told you, don't worry about it. All right? We'll talk about it later. Nucleic acids. Ready? They're polymers. So 3 of these 4 are true monomer-polymer situations. The carbohydrates, right? The proteins, and the nucleic acids. The fats, right? Those don't really behave like monomers and polymers, even though they have a fatty acid building block. Cool? All right. Nucleotides, building blocks of nucleic acids. There's 5 different types, way easier than the amino acids. All right? So they've got a similar thing to the amino acids in that they have a repeating portion, this sugar and this phosphate group. That's the same in every nucleotide. All right? What's different is the is the nitrogenous base, they call it. Okay? This group right here. So, you see at this point, with how popular genetics is, we all know this A, T, C, G, U. Those are important. Your genetic sequence is like AAAT, CCGT, TAG, CTA, right? You know what I'm saying? That's all determined by the nitrogenous base. That determines if it's an A, a C, a T, a G, or a U. All right? The U also is RNA, so it has a different sugar. I'll show you that in a second. Think about it like an amino acid. This is different between each type, but the sugar phosphate, those are the same. Are we cool with that? I'm going to call that good. Okay. nucleotides made up of nitrogenous base, all right, a sugar, and a phosphate group. Now, I've told you I'm not going to be worrying a lot about carbon numbering. I just think it's not, for biology, it's not a great use of our time. But when we're talking about organic molecules, sometimes you want to talk about something attached to a specific carbon. So in a carbon-based molecule, you give the carbons numbers. In this sugar here, the carbons are carbon 1, 2, 3, 4, and 5. All right? Carbon 3 has a hydroxyl group coming off of it. Carbon 5 has the phosphate group coming off of it, all right, in the sugar. This is going to be important because the dehydration synthesis that makes covalent bonds, making a long polynucleotide or nucleic acid, is going to involve joining the 5' and the 3' carbon sort of together. All right? So you're good. This is the only time you're going to need to know the carbon number, the 5' versus the 3'. All right? The 5' has the phosphate group, the 3' has the hydroxyl group. Rather than doing a whole 30-minute lecture about here's how the rules for numbering carbons, I just wanted to talk about the one time you need to know it in general biology. Yeah? Mm-hmm. Hydroxyl. Yep, exactly. Cool. And the really key thing is going to be that energetically you can only add a new nucleotide here, all right, with the enzymes that we have. So when we're growing a polynucleotide, you're going to be adding new nucleotides to this end. And that's incredibly important, actually. Okay? Cool. Don't worry, I got a lot more reflection on that, a lot more information, a lot of repetition. You know, I love my repetition. The nitrogenous bases. There are 5 different nitrogenous bases, okay? Remember, this is the nitrogenous base, this part right here. It's got a bunch of nitrogen in it, okay? We've got cytosine, thymine, uracil, adenine, guanine, CTUAG. Those are the 5, okay? They are broken down into 2 groups, the pyrimidines and the purines. The purines are adenine and guanine. The pyrimidines are cytosine, thymine, and uracil. Pyrimidines have 1 ring in the in the base, nitrogenous base. Purines have 2 rings in the nitrogenous base. It matters in detecting, when the DNA is being replicated and the cell is detecting, did I put the right nucleotides in? This matters a lot, okay? Now, DNA is a double-stranded structure, okay? Held together, each strand is held together by hydrogen bonds that occur between the nitrogenous bases, all right? And it's always a purine interacting with a pyrimidine, okay? Or a pyrimidine interacting with a purine, the statement's the same. It's always a purine on one side and a pyrimidine on the other side. And think about that, right? If my arms are the 2 strands and they're set at a certain width, distance from one another, well, if I have a purine and then a pyrimidine, that's 3 total rings. Same thing. But if I had 2 purines, they'd get pushed apart. And if I had 2 pyrimidines, the strands would sort of come together. So the width is always consistent, and it's set by the fact that it's a pyrimidine bound to a purine. That allows the DNA post-replication to be sort of fact-checked. Because if it's like, oh, accidentally added a pyrimidine where there shouldn't be one, or sorry, a purine where there shouldn't be one, the DNA is too wide, let's repair that and fix it. All right? That'll matter later, I promise. Cool. Pentose sugar. There's a 5-carbon sugar, a pentose, in every nucleotide. Now, DNA and RNA are a little different from one another, okay? DNA, or deoxyribonucleic acid, right, is different from RNA, or ribonucleic acid, and one of the most obvious ways is in the sugar. So every nucleotide in DNA is a deoxyribose. There's one less oxygen, deoxy. Every sugar in an RNA is a ribose sugar, where look, the hydroxyl group's maintained there. So deoxyribose is the sugar in DNA. Ribose is the sugar in RNA. Otherwise, they're pretty similar. They're going to be joined together in a series of sugars and phosphates and sugars and phosphates. We cool so far? All right. Now here's a nucleotide polymer. This is a single-stranded nucleotide polymer right here. Sugar phosphate, sugar phosphate, sugar phosphate, sugar phosphate, with a bunch of different nitrogenous bases projecting off. Cool? Very cool. Okay. So the sequence of DNA or RNA is determined by the sequence of bases. That's what gets read, is the sequence of bases. This here is the backbone. It's repeating, just like in the polypeptides. And we call it the sugar phosphate backbone because it's sugar phosphate, sugar phosphate, sugar phosphate, sugar phosphate. Fair enough? Nothing too wild there. Every piece of information is hopefully accessible. The only problem in summer school is the sheer amount of information coming at you, right? Now, I'm going to put another piece on this, which is that 3', 5' piece, right? The carbons have been numbered here. See at this end of the sugar of the polynucleotide, the 5' carbon is on this end. We call this the 5' end of the polynucleotide, or the nucleic acid, same thing. Okay? We call the other end the 3' end, because on this end, the 3' carbon's projecting off this hydroxyl group. You can only add new nucleotides to the 3' end. So just like, you know how I mentioned very briefly how there's an N terminus and a C terminus on an amino acid, how it has ends that we can identify? The same thing's true with DNA and RNA. The molecule has a 5' end and a 3' end, all right? And you see how they are linked via covalent bonds to this phosphate group between the 5' and the 3' carbons. Cool? All right. All right. Very good. Adenine, thymine, they always interact. Guanine, cytosine, they always interact. All right? The dotted lines indicate hydrogen bonds. And look, you know that these could be hydrogen bonds because there's an oxygen over here. There's a hydrogen right here. There's a nitrogen here. Nitrogen's also super electronegative. So that's going to make a partial positive charge here, a partial negative charge here. Very polar, hydrogen bond. All right? A to T, C to G. All right? The way to remember that is there's only one word that you can make out of the letters A, T, G, C, and that's at, A to T. And once you know A binds T, you know G must bind C. I don't know. That's helpful. Hey, check this out. There are 3 hydrogen bonds between guanine and cytosine. Technically, that makes this interaction more stable than this interaction, which means that DNA that's enriched in a bunch of G's and C's is actually more stable than DNA enriched in a bunch of A's and T's. Does that matter in Bio 105? No way that matters in Bio 105. But maybe if you continue into genetics, it'll matter. DNA is double-stranded inside the cell. All right? There it is. There's the molecule. It's like the most famous thing. There's 2 things you know coming into a biology class. You know micropipetter goes into tube and you look like this, all right? And you know DNA spins on all the computer monitors. And that's how you know you're working in a lab, right? Like in a movie, okay? There's the DNA double helix. What is it? Well, it's 2 polynucleotides, right? Sugar phosphate backbone with the nitrogenous bases projecting off, and the nitrogenous bases associate with hydrogen bonds. And that holds the double-stranded structure together, okay? The sequence of nucleotides gets read off of one of these strands and used to direct proteins, protein synthesis. Just that easy. Look at this molecule. There's a 5' end, there's a 3' end, there's a 5' end, there's a 3' end. There's the bases interacting via hydrogen bonds. A to T, C to G, every time. Cool? All right. Now, we call the DNA strand anti-parallel. I think that this slide is an evil slide. It is bad, it's a cursed slide. Because just looking at this and the text DNA strand is anti-parallel, well, what do you think that means? Well, I look at this and I say, like, it goes TGCA and then TGCA. So the sequence in one direction, one strand of DNA, is the sequence in the opposite direction. No, that is not what it means at all. And that statement is not true, okay? If that was true, then each single strand of DNA would be exactly the same, right? And actually, they can be totally different, and sometimes there'll be one gene here and a completely different gene here. So you get to pack more information in, right? So that's really important. What we mean when we say the DNA strand is anti-parallel is that the 5' and 3' ends are opposite one another, okay? So this strand's 5' end is here. This strand's 3' end is over here. Do you see what I mean? So it is not that the strands are identical in their sequence, but opposite. In fact, when I first came here to teach this course, and the first time I taught this course, I was a little overwhelmed. And I looked at this slide and I was like, DNA strands are anti-parallel. So if you look at one in one direction, then it's the same in the other direction. I told an entire class of like 70, 80 students that. Then I had to come back the next day and apologize and correct myself, you know? And I just finished 6 years of a PhD in plant physiology. And I was still wrong about anti-parallel DNA. Let's prove it right now, okay? 5', 3', 3', 5'. Here's a sequence. A, C, G, T, A, C, G. The complementary sequence will be C, G, T, A, C, G, T, T, T. That's not the same at all, right? If we flipped this over and looked at it, you know, A is not C, G is not A. See what I mean? Cool. So the anti-parallel thing relates entirely to the 3' and the 5' ends. Good? And I just want to emphasize again that DNA synthesis must be in the 5' to 3' direction, meaning you can only add new nucleotides to the 3' end of one of these strands. It's going to be so important later when we get into DNA replication. Okay, let's summarize the difference between DNA and RNA. The one thing I haven't talked to you about is that RNA is single-stranded. DNA is double-stranded. All right? They both have a sugar phosphate backbone with nitrogenous bases projecting off. Okay? In DNA, deoxyribose is the sugar. In RNA, ribose is the sugar. It's still a 5-carbon sugar. In DNA, all right, and in RNA, there are 4 nitrogenous bases, but they are different between the two. So, in DNA, you have A, C, G, and T, adenine, cytosine, guanine, and thymine. But in RNA, the bases are adenine, cytosine, guanine, and uracil. All right? So, U right there. Cool? So an RNA nucleotide will look different from a DNA nucleotide. Even if they both have cytosine as the base, they'll have a different sugar. Okay? Now, in RNA, U goes everywhere a T would usually go. So what do I mean? Well, here we go. Let's say that I'm reading a strand of DNA in order to synthesize a strand of RNA. Well, I know I have to synthesize it in the 5' to 3' direction. So the first thing is it's going to be synthesized in this direction. Where there's a G, there's a C. Where there's a C, there's a G. Where there's an A, there's not a T, there's a U. Okay? That's the direction of synthesis, and here's what the RNA would look like. Okay? And when this happens in the cell, basically what goes on is you got double-stranded DNA, and you temporarily separate it, and then you make the complementary RNA and pull that off, allow the DNA to come back together, take that RNA, fix it up a little bit, and then give it to a ribosome to make a protein. I know it's complicated, but don't worry. We got lots of time to talk about it. We still got a whole month together to get sick of these concepts. Last slide. Let's do it. All right. Summary of all the biological macromolecules. It's missing my first, my column titles, but that's okay. Here are the 4 different types of molecules: carbohydrates, lipids, proteins, and fatty acids. Those are the big biological macromolecule categories. These are the monomers, okay, or the subunits, because remember, the fatty acids don't really work like monomers because they don't get joined into a sequence. So in the carbohydrate, the monomer is the monosaccharide. In the lipid, they have a building block that is a fatty acid. In the protein, the monomer is the amino acid. In a nucleic acid, the monomer is the nucleotide. And I know I've been saying polynucleotide a lot. That's another name for a nucleic acid. I like to use it because it's like poly, many nucleotides. All right? So either term is okay. Oh yeah, there's the name for the polymer types: polysaccharide, triacylglycerol, not a true polymer, but the triglyceride, right, for the lipids. Polypeptide is the name for the protein polymer. Polynucleotide is the name for the nucleotide polymer. There it is. And then you do not need to memorize the linkage types. But these are the bond types that hold all of the different subunits together. It's a glycosidic linkage in the carbs. It's an ester linkage that holds the lipids to the glycerols. It's a peptide bond that holds the proteins together. And it's a phosphodiester linkage that holds the dang nucleotides together. All right, cool. They're all covalent bonds right there. All right, folks, that finishes our first PowerPoint. That finishes our first week of Summer Bio 105. So thank you all so much. That is 20% of the course done. All right? Hopefully that makes you feel pretty good. Keep up the good work. Keep showing up. Keep asking me questions if you got them. Recordings, I'll get them all posted by the end of the week. And don't forget those assignments, okay? Those assignments are due. They should show up on your to-do list on Canvas. But otherwise, we're good, I think, right? All right.